So for chapter 6, what we're going to do is we're going to take an in-depth look at all the neurotransmitter systems. Now, not all of them because there are a lot of neurotransmitter systems, but we're going to look at the major ones that you definitely need to understand and know, like the catecholamines, acetylcholine, serotonin, glutamate, GABA. We'll talk about some general principles of neurotransmitter systems, what we need to do in order to identify something as a neurotransmitter, a brief review of synaptic activity, some of the research methods we can use like immunohistochemistry and situ hybridization to be able to categorize and label neurotransmitters or regions in the brain where they may exist. And then we'll go and finish off our discussion by looking more so at the structure and the functions of ionotropic receptors versus metabotropic or G protein coupled receptors. So here we go. So in the last chapter, we looked at how synaptic transmission worked. So what we had was our presynaptic terminal. We had our action potential, which came down to the terminal. The depolarization of the terminal led to voltage-gated calcium channels opening up, allowing for calcium to flow in. When calcium came in, what we had was the activation of the snare protein complexes. That allowed for the vesicles filled with neurotransmitters to fuse with the plasma membrane of the terminal, allowing for neurotransmitter to come out into the synaptic cleft. And then we left our discussion with they would be able to bind to receptors. In this lecture, what we're going to do is we're going to talk more so about what these neurotransmitters are, what receptors they can bind to, and what happens when they bind to the receptors. So a brief review, we have 3 different classes of neurotransmitters that we often think of. We have amino acids, such as glutamate and GABA and glycine, amines, like dopamine, acetylcholine, serotonin, and we also have peptide neurotransmitters or neurohormones. So there are many different classes and types of neurotransmitters that exist within the brain. And so a few ways that we can define particular transmitter systems is we can look at the actual molecule itself, we can look at how it is actually created, how it's packaged, how it's brought back up, and then how it is degraded. And then, of course, we can look at what kind of action does it have. Is it inhibitory or excitatory? Does it work on several different areas of the brain versus one particular area of the brain, etc.? So the first identified neurotransmitter was acetylcholine. And again, as a brief review from last chapter, this was seen through Otto Louis and the frog heart experiment, where he stimulated the vagus nerve and found that the heart in the saline solution with the vagus nerve attached, when he stimulated that vagus nerve, he found the heart rate decreased. When he took that solution around that heart and moved it into a second jar with a non-connected vagus nerve on a second heart, the heart was seen to slow down as well. So one way that we are calling these neurotransmitter systems or these neurotransmitter containing neurons is we'll take their basic name and add an ergic to it. So if it contains acetylcholine, it's a cholinergic neuron. If it contains dopamine, it's a dopaminergic neuron. If it contains norepinephrine, it's a noradrenergic neuron, etc. So first we need to identify what the neurotransmitter is, right? If we're thinking of something as a neurotransmitter, we need to be able to identify it. So there are 3 criteria for neurotransmitters. 1, it has to be synthesized and stored in a presynaptic terminal or in a presynaptic neuron. So when we think about neurotransmitters, we're thinking about chemicals or molecules that are packaged into vesicles that will then be released out here in the extracellular space or in the synaptic cleft, which can then act on receptors on the postsynaptic cell. So the second criteria is it needs to be released by the presynaptic axon terminal upon stimulation. So when an action potential comes down, this neurotransmitter needs to be released from this terminal due to that action. And then the third criterion is when it is experimentally applied, it needs to mimic the postsynaptic cell response produced by the release of neurotransmitter from the presynaptic neuron. So let's say acetylcholine. If the way acetylcholine is packaged and sent out and affecting the receptors, if I were to take acetylcholine separately and inject it onto a cell, would it have the same postsynaptic cell response? So these are the 3 criteria for what makes essentially a neurotransmitter a neurotransmitter. So the first step in confirming that some molecule is indeed a neurotransmitter, we can use immunocytochemistry and in situ hybridization, which are two of the most important techniques to use. And one thing that we can do with these techniques is we can identify areas in the brain or cells within the brain that contain these neurotransmitters. So localizing transmitters and transmitter synthesizing enzymes can be accomplished through the use of immunocytochemistry and immunohistochemistry. So immunocytochemistry is when we anatomically localize particular molecules and we can identify the cells in which those molecules reside. And immunohistochemistry is when we use that same technique and apply it to thin sections of tissue where we can use labeled antibodies to identify the locations of proteins or molecules within those given cells. So what I'm going to show you is just a brief video which gives you a general summary of immunohistochemistry, and it gets you sort of familiar with some of the ideas that we're going to discuss in the next slide on immunohistochemistry.
Antibodies, also called immunoglobulins or IgG molecules, are used by the immune system to attack foreign substances called antigens that enter the body. This property can be used to generate antibodies of clinical interest. For example, if an antigen from human cells is injected into a rabbit, the rabbit's immune system will produce specific antibodies against the antigen. The resulting rabbit antibodies can then be withdrawn, purified, and applied to human tissue specimens to identify the location of the antigen within a cell. Antigens are typically proteins or polysaccharides within or on the surface of cells. Antibodies bind specifically to the antigen that triggered the antibody's production, and as a result, immunohistochemical reactions can be very precise. To identify the location of the bound antibody, and thus the location of the antigen, we can use a procedure called the direct method. In this method, the antibody is attached or conjugated to an indicator molecule. Different indicators include fluorescent molecules, biotin, or in this case, the enzyme horseradish peroxidase, or HRP. In the direct method, the purified conjugated antibody is applied to the tissue and allowed to react with the antigen. Unbound antibody is washed away. When HRP is used as an indicator, the hydrogen peroxide substrate is added to the incubation mixture, as well as a third component called diaminobenzidine, or DAB. When coupled to the HRP peroxide reaction, DAB forms an insoluble polymer that sticks to the adjacent tissue. Other chemicals can intensify the color of the reaction product. For example, adding nickel sulfate to the DAB mixture will turn the precipitate purplish black. The location of the precipitating product marks the location of the antigen in the tissue. In this example, the antibody was used to indicate which cells express a particular antigen. When we use antibodies as probes to detect specific molecules, we frequently want to amplify the signal by using a procedure called the indirect method. In this method, an unlabeled antibody, called the primary antibody, is first added. The primary antibody binds directly to the antigen of interest. Excess antibody is then washed away. In this example, the primary antibody is an IgG molecule made in rabbits that recognizes an antigen we will call antigen X. Thus, the antibody is called rabbit anti-X IgG. In the next step, a linking antibody that recognizes rabbit IgG molecules is added to the mixture. In this case, the linking antibody was produced by injecting rabbit immunoglobulins into another species, such as a goat. The linking antibody thus recognizes rabbit IgG. Note that after binding to the primary antibody, the linking antibody retains one free antigen binding site. The final step of this example of the indirect method uses a complex that consists of the peroxidase enzyme conjugated to an antibody that recognizes the peroxidase as an antigen. Note how the antibodies in the peroxidase-antiperoxidase, or PAP complex, build on each other. The antibodies in this complex come from the same species as the primary antibody, which is rabbit in this case. Therefore, the free ends of the linking antibodies will bind both to the antigen on the tissue as well as the antibodies in the PAP complex. As in the direct method, antibody is washed away and a substrate is added to localize the reaction. The choice of whether to use the direct or indirect method depends on the purpose of the experiment. The direct method entails fewer steps and is less prone to nonspecific background labeling. However, it requires that the primary antibody be specifically labeled. The indirect method is generally more sensitive and is also more versatile because a variety of primary antibodies made in the same animal species can be used with the same conjugated secondary antibody.
So I'm not going to test you heavily on that knowledge really, because when you are working in a lab or if you're using these techniques, you typically will learn them there. But one thing that I will point out is that these techniques are very important, particularly in lab spaces where we're trying to identify the presence or the quantity of protein that are found in either tissue or in brain homogenate. And I use these techniques all the time in Western blotting in my research. So I use mouse models of Alzheimer's disease. So I will take a rabbit anti-mouse antibody as a primary antibody. So that antibody will stick to a particular protein of interest. And then I'll use a label through horseradish peroxidase, goat or donkey anti-rabbit. So I use the indirect method. Will bind to the rabbit anti-mouse antibody, and then I can semi-quantitate how much protein is present in my particular samples. So in immunohistochemistry, if you break it down, it just simply means antigen or antibody based, so based on the immune response. Histo is dealing with tissue, so remember histology is the microscopic study of tissue, and then chemistry, looking at reactions. So stimulation of an immune response by a chemically purified neurotransmitter candidate can be injected under the skin or in the blood in a different species, which creates antibodies against that. Antibodies are then generated, which can then bind tightly to specific sites on the foreign molecule, known as antigens. So in our unknown neurotransmitter, it can bind to the transmitter candidate. We can then chemically tag those specific antibodies. That can then be seen with a microscope or in a particular instrument. And then when those labeled antibodies are applied to a section of tissue, they color just those cells that contain them. So we can use this criteria to demonstrate our transmitter candidate is localized in a particular neuron or in regions of neurons like in the axon terminal. So here's a brief example of a neuron in the cerebral cortex labeled with antibodies that bind to a peptide transmitter. And the fact that it's binding to a peptide transmitter will show that this cell contains that neurotransmitter. And then you could also use multiple antibodies using different colored fluorescent markers, and you can use fluorescence microscopy to be able to look at different types of neurons in the same region of the brain, just depending on your excitation and emission wavelengths and overlapping these signals based on where you're coloring. So this cell might contain something like GABA versus this cell which contains glutamate. And so you can fluoresce at the same exact time and see the presence of these neurons. So another way to localize transmitters is through the process of in situ or in situ hybridization. And this is useful for confirming that a cell actually synthesizes a particular protein or a particular peptide. So if you recall from chapter 2, yes, we're going back to chapter 2, proteins are assembled by ribosomes according to specific mRNA molecules, and this is the process of translation. Remember, transcription is where we took the DNA and created an mRNA transcript. Translation is where we take that mRNA transcript and we synthesize a protein. So unique mRNA molecules exist for each polypeptide that is synthesized. So there are 4 different nucleic acids linked together, A, U, C, and G, and each of these nucleic acids binds most tightly to one other complementary nucleic acid, A to U and C to G. Now, if you know a sequence of nucleic acids that exists in a particular strand of mRNA, then you can construct a complementary strand. So for instance, if you had a strand that was A, U, C, G, C, U, A, then you can create a complementary strand that would bind to that. It would be U, A, G, C, G, A, U. And that's what in situ hybridization does, is we create a complementary strand to a known sequence of nucleic acids, and this is what's known as our probe. And the process by which the probe binds to that mRNA molecule is known as hybridization. So probes have the ability to be chemically tagged. And remember, the probe is the sequence that we are creating that will be able to bind to our mRNA single-stranded molecule. So we can make them radioactive, and we can detect them by laying the brain tissue on a sheet of special film that's very sensitive to radioactive emissions in the film. And after we expose the tissue and develop it, we can see these negative images of radioactive cells visible as clusters of white dots. And this is what's known as autoradiography, where we can view the distribution of radioactivity that's present. So here in this diagram, we have a strand of mRNA in a neuron. And what we do is we take our probe, which is the complementary strand. So we radioactively label it with the proper sequence of the complementary nucleic acids so that it fits together. And then what we can do is lay it down on a piece of brain tissue, and we'll actually be able to see where that mRNA lies and in what particular cells. So if you didn't want to do radiography, or if you don't want to mess with radioactive emissions or radiation, you could also label the probes with fluorescent molecules to view under a fluorescence microscope, and this is what's known as FISH, or fluorescence in situ hybridization.
Given time, only some of those genes are even being expressed. When a segment of DNA or a gene is on or being expressed in a cell, it's being transcribed into small strands of RNA, which are what provide the blueprint for protein. When a gene is off, no RNA is being transcribed and thus no protein produced. A gene could be off because that protein is no longer needed, or maybe it's not needed in that particular part of the body. During embryonic development, genes are constantly being turned on and off, at different times and in different places along the body, a perfectly timed orchestra of genetic mechanisms to go from this to this. So what we need is a way to look inside the embryo at various points in gestation to look for the presence of RNA. It isn't foolproof, but the presence of RNA for a gene in a certain part of the body is a pretty good indicator that that gene is doing something there. To do this, you need a specific probe. Here's where we make use of the fact that RNA, unlike DNA, is single-stranded. A probe is just another single strand, synthesized in a laboratory, which is complementary to the RNA of the gene that you're looking for. Here's a mouse embryo taken after 15.5 days of embryonic development, essentially frozen in time. When the embryo is put into a solution containing a probe specific to the RNA of the gene of interest, the probe finds all the places in that embryo where the specific RNA is present and hybridizes to it. Hence the term, in situ hybridization. Lastly, the probe must be chemically labeled with something, whether it's a radioactive label, a label that fluoresces under UV light, or a label that can be detected by an antibody, such as this one. With a label to tell you where the probe has hybridized, you can look at the embryo under a microscope and see where, if anywhere, that gene is turned on in the embryo. And so you're one step closer to learning what that gene is doing.
So in order to localize proteins, we can use immunocytochemistry or immunohistochemistry to view the location of specific molecules, including proteins, in different sections of brain tissue. In situ hybridization is a method for localizing, again, mRNA transcripts for proteins. And together, these methods help us to see whether a neuron contains and or synthesizes a transmitter candidate and molecules associated with that transmitter. So we can probe and look for synthesizing enzymes, the neurotransmitter itself, etc. So the transmitter candidate is synthesized and localized in the terminal, and it's released upon stimulation. So we can then take samples of the fluid bathing that synaptic target. And this is the basis of Otto Loewi's experiment, where there is some chemical that is released upon stimulation that we can then capture. So the central nervous system contains a diverse mixture of synapses that use different neurotransmitters, depending on different neurotransmitter-containing cells. So we can use brain slices as a model. So we can keep these alive in vitro, so outside of the brain, and we can stimulate synapses bathed in high potassium concentration solutions, and we can collect and measure released chemicals. So one thing to note, and one thing that is of importance, is that calcium must be present in the solution as well. So remember snare proteins. Snare proteins, the calcium sensor synaptotagmin, must be present in order for vesicle fusion to occur. And we're getting sophisticated in science, and new methods like optogenetics allow us to activate just a particular type of synapse at a time, where we can actually say, okay, neurons that have channel rhodopsin or halorhodopsin in the presence of blue or yellow light respectively will only be the ones that are going to be activated. So studying synaptic mimicry, one of the qualifying conditions for neurotransmitter identification is that molecules evoke the same response as a neurotransmitter. So this is the third criterion for identifying if something is a neurotransmitter. The same response must be evoked as that produced by the neurotransmitter released from the presynaptic terminal. So if released from the presynaptic terminal, this neurotransmitter has an excitatory effect, if I were to take that neurotransmitter and apply it directly to the postsynaptic cell, would it have an excitatory effect as well? So what we can do is we can use this cool technique known as microiontophoresis, and we can assess postsynaptic actions of a cell. So what we do is we fill our glass pipette over here with our solution that contains a possible neurotransmitter and position it next to the postsynaptic membrane while measuring the membrane potential with a microelectrode. So if we apply the molecule and it causes some electrophysiological change, and the other criterion are met when we are trying to identify whether it's a neurotransmitter, then the molecule under study and the transmitter are usually considered to be the same chemical. That makes sense, right? So if you are looking at this unknown chemical and you think it's a particular neurotransmitter, and you apply it postsynaptically and it has the same effect as it does normally, then you can pretty much be safe in assuming that it is going to be the same neurotransmitter. So here's your drug-containing micropipette. You apply the drug, you stimulate the axon, you record the membrane potential, and you see whether or not there is a similar effect. So each of the different receptors a neurotransmitter binds to are different receptor subtypes. And so what we can do is we can use neuropharmacological analyses to look at classes of compounds known as agonists and antagonists. Agonists mimic the effects of the neurotransmitter, and antagonists block the effects. So we covered that briefly in last chapter. And so there are three approaches to studying receptors and receptor subtypes specifically. You can use drugs, and neuropharmacological analyses are looking at agonists and antagonists to be able to block and or stimulate receptors. You can use ligand binding methods, which are just simply compounds that will be able to bind. And you can look at the molecular analyses of receptor proteins. So you can actually look at the molecular level what amino acids are present in the channel pore or lining the receptor membranes, and you can identify at a chemical level and a molecular level what's interacting where, what kind of regions are being moved or conformational changes are occurring, etc. So our first little step in our whole ride throughout the neurotransmitter systems will be acetylcholine. Here we're going to focus on its receptor, and then I believe in the next slide we'll go through glutamatergic receptors. But acetylcholine will be our first big major neurotransmitter that we go through. So acetylcholine receptors, you have two different types. You have muscarinic, which is dealing with the agonist muscarine, and then we have nicotinic acetylcholine receptors dealing with its agonist nicotine. So nicotinic acetylcholine receptors are found in skeletal muscle. Muscarinic receptors are found in the heart. Now these receptors also exist in the brain, so don't think that this is just exclusive to these sections of the body. And some neurons have both types of receptors present. Now what is different between these is nicotinic acetylcholine receptors are ionotropic, whereas muscarinic receptors are G protein coupled. So we can use selective antagonists to distinguish receptor subtypes, because certain antagonists will only work at particular receptor subtypes. So both of these receptors are acetylcholine receptors, meaning acetylcholine will bind and work at both of these. However, other things like agonists or antagonists will only work at a particular subtype. So curare from this South American arrowtip poisons can inhibit actions of acetylcholine at nicotinic receptors. This causes paralysis due to the fact that these nicotinic acetylcholine receptors are found in skeletal muscles. Atropine, which is derived from the belladonna plant, also known as deadly nightshade, antagonizes acetylcholine specifically at muscarinic receptors. So note that atropine delivered onto an acetylcholine receptor will specifically block the effects on a muscarinic receptor, but not nicotinic. Nicotine, when applied to a receptor, an acetylcholine receptor specifically, will bind to the nicotinic receptor, but not the muscarinic. And acetylcholine, as these are subtypes of the acetylcholine receptor, will bind to both. There are 3 different subtypes of glutamate receptors. You have AMPA, NMDA, and kainate receptors. Each of these are named for a different chemical agonist, meaning that a specific chemical that is able to bind and activate. AMPA stands for alpha-amino-3-hydroxy-5-methyl-4-isoxazole propionate. Conveniently, we just call it AMPA. NMDA stands for N-methyl deaspartate. Now note, AMPA only acts at the AMPA receptor. NMDA acts only at the NMDA receptor. And kainate acts only on the kainate receptor. They are subtypes. They are specific to those types. However, glutamate will activate all 3. So here's just a brief table, 6.1 from your book, showing the different neurotransmitters, some different subtypes that exist in their receptor systems. So acetylcholine has 2 receptor subtypes, norepinephrine has 2 receptor subtypes, glutamate has 3. Right here is not listed the kainate receptor. GABA, primarily 2, ATP, adenosine. And then it lists their agonists and antagonists. Now, by no means are you going to need to memorize these, but some of the bigger ones are important to know, especially as you continue your career in neuroscience: nicotine and muscarine, AMPA and NMDA, propranolol, phenoxybenzamine, curare and atropine, bicuculin, etc. And in a lot of different pharmacological papers, or if you're looking at a lot of in vitro cell studies, a lot of these antagonists and agonists are given in cell culture to be able to stimulate or antagonize different cellular effects, to be able to test the effects of drugs or different compounds on cells. So in order to study receptors, we can use what are known as ligand binding methods, where we can look at identifying natural receptors using radioactively labeled ligands. So these are radioactively labeled drugs that should bind tightly to particular receptors. And then what we can do is we can actually label these sites and see where that binding has occurred. So an example are opiate receptors, and opiates are commonly abused and medically important. They're a broader class of opiate-like chemicals that have numerous physiological effects, including pain relief, euphoria, depressed breathing, and constipation. Mainly pain relief and euphoria is why they are so often medically abused. Now, radioactive drugs can label specific sites on the membranes of some but not all neurons in the brain, specifically the ones that are going to be engaged in binding with these opioids. So we can search for endogenous opioids or endorphins. These are the naturally occurring neurotransmitters that act on these given receptors. And so I try to identify these opioid neurotransmitters, or these enkephalins, through use of radioactive labeling and be able to identify where in the body, or in the brain rather, these receptors are found. So looking at in the molecular analysis for receptor protein classes, we can identify receptors as being transmitter-gated ion channels or as G-protein coupled receptors. And some neurotransmitters can have both. Some neurotransmitters can act on ionotropic receptors, and some can work on G-protein coupled receptors. So one instance is GABA, or gamma-aminobutyric acid, which is the major inhibitory neurotransmitter in the nervous system. It has a subclass of transmitter-gated ion channels known as GABA-A receptors, and then G-protein coupled receptors, GABA-B. So GABA receptors under the transmitter-gated ion channels contain 5 subunits, each made with 6 different subunit polypeptides. So there's numerous potential subtypes for GABA-A receptors alone. But not all of those are utilized. However, it does allow you to understand the diversity of receptor subtypes that can exist in a given section. So again, the major neurotransmitters include amino acids, amines, and peptides. Dale's principle is that a neuron has only one neurotransmitter. However, a lot of peptide-containing neurons, which co-release a peptide along with a neurotransmitter, violate this principle, because the co-release of a peptide and an amino acid or an amine is in direct conflict with this principle. Now, not every neuron contains both peptides and neurotransmitters, but you're never going to have a neuron that is firing or releasing two separate neurotransmitter classes. So you're not going to have a dopaminergic neuron that also releases acetylcholine or serotonin. So co-transmitters are what are referred to as two or more transmitters being released from one single nerve terminal. So you can have an amino acid or amine plus a peptide, and that's usually the case. Another instance of co-transmitters are things like zinc, so glutinergic neurons, which exist in the cortex. So glutinergic neurons. These are important for particularly for learning and memory, but these are a combination of glutamate as a primary neurotransmitter and then the ion zinc actually being co-released with that glutamate. Just going to grab some water. So most of the known neurotransmitters fit pretty nicely into your amino acids, your amines, and your peptides. A few other non-classical neurotransmitters have been found though, and these are neurotransmitter examples that still behave like neurotransmitters in some fashion, but they're non-classical, such as lipids, which diffuse through the plasma membrane, gases, which also diffuse through the plasma membrane, and others including ATP. So you can classify neurons based on which neurotransmitters they release and what's inside them that are used to actually synthesize these neurotransmitters, what are used to store the neurotransmitters, and what kind of enzymes or chemicals are used to degrade these neurotransmitters. All right. So the first neurotransmitter that was identified and the first one that we will investigate is acetylcholine. And acetylcholine is found at the neuromuscular junction, as we talked about in last chapter, but it's also found within the brain, particularly in the hippocampus. Now, in each of these neurotransmitter systems, we're going to highlight how it's synthesized and how it's broken down. So, acetylcholine is synthesized in the presynaptic terminals by an enzyme known as choline acetyltransferase. This is only present in acetylcholine neurons. So, if you were trying to find where these acetylcholine neurons lie in the brain, you might probe for this enzyme right here, because anywhere in the brain where it has this appearing is going to be an indication that, hey, that neuron is important for synthesizing that neurotransmitter. So, your precursors are acetyl-CoA plus choline with the enzyme choline acetyltransferase will yield acetylcholine plus CoA. So your rate-limiting step is the part of the neurotransmitter synthesis that will determine the amount of neurotransmitter created. And usually if teaching in person, I would ask you what a rate-limiting step is and try to get some class discussion on that. But obviously, since I'm talking to a computer, that doesn't go over that well. But the transport of choline into the terminal through choline transporters is actually the rate-limiting step. If you only have so much choline, imagine you only have 5 units of choline. I'm not going to be able to produce 15 units of acetylcholine from 5 units of choline, right? So the rate-limiting step in this process is the transport of choline. So we can only produce so much acetylcholine based on how much choline we have. Acetylcholine, once it is created in the terminal, remember it's created inside the terminal, is going to be transported into vesicles via the acetylcholine transporter. So acetylcholine transporters are going to take that acetylcholine that we made, and it's going to put it inside the vesicles that lie within the terminal. So acetylcholine will then be packaged into the vesicle, and then through exocytosis, acetylcholine will be released out into the terminal. Now, acetylcholine needs to also be broken down. It can't just continually remain at the level of the neuromuscular junction or in the synapses within the brain. So the enzyme that is important for breaking down acetylcholine is known as acetylcholinesterase. This is ACHE. So acetylcholinesterase breaks down acetylcholine into acetic acid and into choline. Now, what happens with this choline? Well, this choline can actually be brought back into the presynaptic neuron through choline transporters. And that choline will go in and allow for acetylcholine to be produced again. There is no reuptake of acetylcholine. None. You don't have acetylcholine released out here and then acetylcholine as an entire molecule being reabsorbed or reuptake or go through reuptake. That doesn't happen. Acetylcholine is broken down in the synaptic cleft out here by acetylcholinesterase. So here's the brief life cycle of acetylcholine. You have choline in the presynaptic terminal and acetyl-CoA through choline acetyltransferase, allowing for acetylcholine to be produced. Acetylcholine, chilling out here in the presynaptic terminal, will be placed into vesicles through the acetylcholine transporter. Through the process of exocytosis, acetylcholine will be released out into the cleft where it can bind and activate different receptors wherever you happen to be within the brain. Acetylcholinesterase will break acetylcholine down inside the cleft into choline and acetic acid. Choline will then be transported back into the presynaptic cell by choline transporters, allowing for the cycle to begin again. Now, inhibition of acetylcholinesterase prevents acetylcholine from being broken down. That can then disrupt transmission at numerous cholinergic synapses that lie in the skeletal muscles, the heart muscles, as well as in the brain. So acute effects are marked decreases in heart rate and blood pressure. And irreversible inhibition will lead to death, typically resulting from respiratory paralysis. And so here are a few of the different classes of chemical warfare nerve agents and how they are inhibiting and effects on the body. The next class of neurotransmitters are the catecholamines. These are catecholaminergic neurons. These are involved in movement, mood, attention, and visceral function, where one of the best well-known ones is dopamine. Now, why are the catecholamines called the catecholamines? The reason for this is that they contain a catechol group, which is this benzene ring with 2 hydroxyl groups, and they contain an amine group. So tyrosine is an amino acid, and this is the precursor for all 3 amine neurotransmitters. These are the catecholamines: dopamine, norepinephrine, and epinephrine. Norepinephrine and epinephrine are also referred to as noradrenaline and adrenaline, depending on where you're at in the world. So tyrosine will then be converted to L-DOPA by the enzyme tyrosine hydroxylase. Tyrosine hydroxylase is the rate-limiting enzyme. Now, this rate-limiting enzyme can be regulated by the amount of neurotransmitter in the terminal, the amount that the cell is firing, and other second messenger or enzyme messenger activities. The rate-limiting step is here, the conversion of tyrosine into L-DOPA. If you're only going so quickly, or if you only have so much of this enzyme, then you're only going to get so much of dopamine, norepinephrine, or epinephrine. So the synthesis of these neurotransmitters occurs inside the presynaptic terminal. So again, one of the really big key issues in neuroscience is that all these small neurotransmitters are going to be synthesized directly at the level where they're going to be released, so specifically in the presynaptic terminal. So the enzyme tyrosine hydroxylase is the rate-limiting step in the process of catecholamine synthesis. And catecholamines are dopamine, norepinephrine, epinephrine. Tyrosine hydroxylase activity can be regulated by numerous signals that lie in the cytosol of the axon terminal. Because remember, dopamine, norepinephrine, epinephrine are going to be synthesized inside the axon terminal. So decreased catecholamine release by the axon terminal can cause the catecholamine concentration in the cytosol to rise. As this rises, you don't need to keep creating more and more catecholamines. Therefore, through end-product inhibition, this inhibits the enzyme tyrosine hydroxylase. Now, on the other hand, during periods when the catecholamines are released at a very, very high rate, the elevation in internal calcium levels that accompanies those neurotransmitter releases can trigger an increase in the activity of that enzyme, so that transmitter supply keeps up with the demand. So prolonged periods of stimulation can actually cause the synthesis of more mRNA that codes for the enzyme to be created. Now, going specifically into each of the catecholamines, dopaminergic neurons, these are neurons that contain dopamine. These neurons do not have the enzymes necessary to create norepinephrine. They are dopaminergic neurons. Dopamine is transported into vesicles through the vesicular monoamine transporter, also known as VMAT. And there are two main subclasses of dopamine receptors. So you technically have 5 types, D1, D2, D3, D4, D5. But they're combined into 2 different groups. You have the D1-like group and the D2-like group. The D1-like group mainly has excitatory effects, whereas the D2-like group mainly has inhibitory effects. So dopamine receptors are G-protein coupled or metabotropic, meaning that when dopamine binds to a particular receptor, a G-protein will be activated, allowing for an enzyme to be activated or inhibited, and that will cause extracellular or intracellular effects at the level of the postsynaptic neuron. So here you have tyrosine, which is your rate-limiting step, tyrosine to L-DOPA, this being through tyrosine hydroxylase, this being your rate-limiting enzyme and your rate-limiting step, DOPA to dopamine. Dopamine will be packaged into vesicles through VMAT. Through the process of exocytosis, you'll have vesicular fusion occur, dopamine will be released, allowing it to interact with numerous different receptor subtypes to either have an excitatory or an inhibitory effect, depending on the type of receptor that it binds to. Inactivation of dopamine occurs through 2 different methods, through reuptake, through the dopamine active transporter known as DAT, D-A-T, or it can be broken down through enzymes. Now, dopamine can be broken down in 2 separate places. It can be broken down via catechol-methyltransferase that will occur in the synaptic cleft and the terminal itself. So in the terminal, referring to dopamine being brought back up into the terminal to then be destroyed. Or it can be destroyed by monoamine oxidase, specifically in the presynaptic terminal. Norepinephrine contains the enzyme dopamine beta hydroxylase. Dopamine beta hydroxylase is an enzyme that is going to cause dopamine to turn into norepinephrine. Now, what's interesting about norepinephrine is that this is located within the synaptic vesicles and not the cytosol. So you have your reaction that forms dopamine here. Dopamine beta hydroxylase is going to occur here within the synaptic vesicles, allowing for norepinephrine to be synthesized directly in the vesicles. So in noradrenergic axon terminals, dopamine is transported into vesicles through VMAT, and in there, it is then made into norepinephrine. There are 2 different subclasses of norepinephrine receptors. You have alpha and beta. The alpha family of receptors are mainly inhibitory, and the beta family of receptors are mainly excitatory, meaning that if norepinephrine is given, or if norepinephrine goes onto a postsynaptic cell and there are alpha receptors, then there are going to be inhibitory roles. If norepinephrine binds to a beta family of receptors, it's going to have an excitatory effect. Now, the way that norepinephrine can be inactivated, it includes the reuptake, meaning that norepinephrine transporters are going to take up norepinephrine from the synaptic cleft and remove it from the cleft. And it can be broken down in the cleft or presynaptic terminal in the same fashion as dopamine. So catechol-methyltransferase, that can occur within the cleft or the terminal, and then monoamine oxidase, which can occur within the terminal. So the last catecholamine is epinephrine. Now, these are known as adrenergic neurons, and these adrenergic neurons contain phentolamine N-methyltransferase, or PNMT. Now, PNMT exists within the cytosol. So this is like a roller coaster for epinephrine-containing neurons, because what has to happen is tyrosine, through tyrosine hydroxylase, will go into L-DOPA. Dopa decarboxylase is the enzyme that creates dopamine. Dopamine will be placed into a vesicle. Now, in dopamine beta hydroxylase will turn that dopamine into norepinephrine. Now, norepinephrine must actually be secreted and released back into the cytosol, where phentolamine N-methyltransferase, or PNMT, resides. This will then lead to the synthesis of epinephrine. Epinephrine will then be placed and transported into vesicles. And then through the process of exocytosis, it can be released. So epinephrine acts not only directly in a synaptic fashion, but also as a hormone when it's released by the adrenal gland into the bloodstream. And this helps to coordinate our visceral responses, particularly in the fight or flight system. So catecholamine degradation can be affected by different classes of drugs, particularly cocaine and amphetamine. So the way that cocaine and amphetamine work is that they block the uptake of catecholamines, and they prolong their actions in the neurotransmitters in the cleft. So cocaine and amphetamine block the ability of catecholamines to actually be brought back up. So amphetamines enter the dopamine nerve terminals by the dopamine active transporters and disrupt the actions of the vesicular monoamine transporter, which essentially cause neurotransmitter to leak out of the vesicles. Now, as that dopamine concentration increases in the cytoplasm, it can trigger a reverse transport of dopamine by those dopamine active transporters and actually push a lot of that dopamine back out into the extracellular fluid. This causes a massive increase in synaptic dopamine concentrations, and then you have a lot more activation of these receptors. Now, this essentially causes rewarding and addictive actions, primarily as a result of the fact that these dopamine active transporters are in reverse swing, and you have such an increase in the rise of dopamine levels occurring in the presynaptic or in the synaptic cleft. Serotonin is an amine neurotransmitter. It's derived from the amino acid tryptophan, right here. Serotonin plays roles in regulations of mood, emotional behavior, and sleep. And a particular class of antidepressants known as SSRIs are selective serotonin reuptake inhibitors, whereby they are preventing serotonin from actually being brought back up. So they're allowing for serotonin to stay in the synaptic cleft for longer periods. Now right here is the synthesis of serotonin. Tryptophan turns into 5-hydroxytryptophan through the enzyme tryptophan hydroxylase, which is the rate-limiting enzyme. 5-hydroxytryptophan through the enzyme 5-HTP decarboxylase will turn 5-HTP into serotonin, or 5-hydroxytryptamine. In serotonin neurotransmitters, there are at least 15 different receptor subtypes. So this is a huge number of subtypes. And the most well-characterized are 5-HT1A, which is mainly inhibitory, and then you have 5-HT2A receptors, which are mainly excitatory. So again, this is one big common idea in neurotransmitter systems is whatever receptor your neurotransmitter binds to, that is going to be specifically what kind of effects are going to be present in the nervous system. So the amino acid tryptophan is the precursor for serotonin. Tryptophan hydroxylase is the rate-limiting enzyme, and this rate-limiting enzyme activity can be regulated by numerous things. So it can be regulated by the amount of tryptophan in the extracellular fluid that is present. It can be regulated by the amount of serotonin that is actually present here in the nerve terminal, by the amount of activity that's going on, and then other messenger activities that may be occurring inside the terminal. Transport of serotonin goes into vesicles through the vesicular monoamine active transporter. Reuptake occurs via serotonin transporters. And then breakdown of serotonin only occurs in the presynaptic terminal via the enzyme monoamine oxidase. So differences among amino acid neurons are quantitative and not qualitative. So glutamic acid decarboxylase is a key enzyme in GABA synthesis. So this is a good marker for GABAergic neurons because ideally glutamic acid decarboxylase, if it is creating GABA, should only be present in GABA-containing neurons. So GABAergic neurons are major sources of synaptic inhibition in the central nervous system. So trying to find these amino acidergic neurons, including GABA, you need to be very specific in trying to localize where that neurotransmitter lies. So here are 3 of the amino acid neurotransmitters: glutamate, glycine, and GABA. GABA and glycine are primarily inhibitory. Glutamate is excitatory. So here's a plus, a minus, and a minus. So when I say amino acid neurotransmitters, these are neurotransmitters which are also basic amino acids. So glutamate and glycine are of the 20 amino acids, and then glutamic acid, decarboxylase, if you just take away this carboxyl group, or carboxylic acid group from glutamate, that turns into GABA. So glutamate, the major excitatory neurotransmitter in the nervous system, is synthesized from the other amino acid, glutamine, via the enzyme glutaminase. So glutamine will turn into glutamate through the enzyme glutaminase in the presynaptic neuron. So remember, our big idea here is that these neurotransmitters are being synthesized at the level of the terminal. The transport of glutamate into vesicles occurs through V-GLUT, or the vesicular glutamate transporter. Now, after the receptor activation has occurred, the action is terminated through uptake into glial cells. And this is through glutamatergic transporters, or glutamate transporters. Now, what happens to glutamate inside the glial cell here is you have a breakdown of that glutamate through glutamine synthetase, and that turns the glutamate into glutamine. Now, that glutamine diffuses across the membrane back into the presynaptic terminal, so you can have glutaminase active again to convert glutamine to glutamate. So the general process of glutamate is glutamine through glutaminase will turn to glutamate. Glutamate will flow into the vesicles through transport via the vesicular glutamate transporters. You have snare protein complexes, which will allow for vesicular fusion. Glutamate will be released. They can act on metabotropic glutamate receptors or ionotropic glutamate receptors, NMDA or AMPA. Once that glutamate diffuses, is removed through glial cells. The glia will transport glutamate through glutamate transporters, break down that glutamate through glutamine synthetase into glutamine, and then that glutamine will be transported back to the presynaptic terminal where the process can start again. GABA is a byproduct or a product from glutamic acid decarboxylase from glutamate. So the major inhibitory neurotransmitter of the nervous system is actually a product of enzymatic activity done through glutamate, or done on glutamate. So this is synthesized from glutamate by the enzyme glutamic acid decarboxylase. So essentially what you're doing in this chemical reaction is you are decarboxylating the glutamate. So this is transported into vesicles via the vesicular GABA transporter. Uptake of GABA occurs into the glial cells by a GABA transporter, which is present here. So you have a vesicular GABA transporter which places GABA into vesicles. GABA will be released. It can act upon its receptors here on the postsynaptic neuron. Once GABA is present, it can be transported into glial cells through GABA transporters. And what happens is GABA is broken down into glutamate by the mitochondria that are found within the glial cells. Now from that point, you have glutamate as a product. This will undergo transformation to glutamine through glutamine synthetase. Then glutamine will be transported here to begin this whole process. So in a GABAergic neuron, glutaminase will take glutamine to glutamate. Glutamic acid decarboxylase will turn glutamate into GABA, and then GABA can then go into its vesicle and be released. So if you were trying to find GABAergic neurons, you wouldn't want to use the enzyme glutaminase because it would also give you glutamatergic neurons. So glutamic acid decarboxylase is the enzyme, which is a protein, you would want to use to actually be able to say, okay, where are we in the brain? What kind of cells contain GABA? Okay. GABA is the major source of synaptic inhibition that lies within the nervous system. So here are some other neurotransmitter candidates and intracellular messengers that are sort of in the non-classical division. So ATP excites some neurons and it binds and has its activity at the level of purinergic receptors. So these are often packaged in vesicles along other neurotransmitters, meaning that they are co-transmitters with numerous other neurotransmitters. And they're degraded by extracellular enzymes, and ATP is specifically degraded by extracellular enzyme yielding adenosine. And as we also talk about these other non-classical neurotransmitters, we see examples of them being retrograde messengers, meaning that, such as the endocannabinoids and nitric oxide, they act in a reverse fashion by being released from the postsynaptic cell and actually affecting the presynaptic cell. So endogenous cannabinoids, also known as endocannabinoids, are small lipid molecules. Now what happens specifically with these molecules is that they engage in retrograde neurotransmission, where they serve as feedback to regulate neurotransmission in a presynaptic cell. So THC, tetrahydrocannabinol, is able to bind to specific G-protein coupled cannabinoid receptors, and these depend on the certain subclass, CB1 or CB2 receptors. CB1 receptors reside in the brain, whereas CB2 receptors are found in immune tissues elsewhere in the body. So you can find these in particular motor control areas, the cerebral cortex, and different pain pathways. So THC-like neurotransmitters are known as endocannabinoids. They are endogenous cannabinoids, and two of the major cannabinoids in the body are anandamide and arachidonoglycerol, or 2-AG. So endocannabinoids work through the idea of affecting calcium channels. So voltage-gated calcium channels open in the postsynaptic neuron through vigorous action potential firing, and this increase in intracellular calcium rises, which stimulates the synthesis of endocannabinoid molecules. Now, endocannabinoid molecules are not packaged in vesicles. They're manufactured pretty rapidly and on demand. They're very small and they're permeable to the membrane, so they can diffuse through the membrane. Binding selectively to the CB1 receptor that lies on the presynaptic neuron, the main effect is to reduce the opening of presynaptic calcium channels. Now, if we are preventing or reducing the opening of calcium channels on the presynaptic terminal, what happens? Well, because you know that calcium is important for snare protein activation and for vesicular fusion, this impairs the ability, right here, this impairs the ability of the presynaptic terminal to release neurotransmitter. So this suppresses either an inhibitory or an excitatory drive onto the neuron. Again, depending on what type of neuron has the CB1 receptor, you're going to either suppress the inhibitory action if it was releasing inhibitory neurotransmitter, or you're going to be suppressing excitatory actions if it was releasing excitatory neurotransmitters. Nitric oxide is an example of a retrograde molecule. This means that it acts in a reverse fashion, going from the postsynaptic up back to the presynaptic terminal. This is a gaseous molecule, so it's a good example of a non-classical neurotransmitter. It's synthesized from the amino acid arginine and has powerful biological effects, particularly in the regulation of blood flow. So it's very small and it's membrane permeable. It's a gas, so it has the ability to diffuse freely, but it does break down very rapidly. Now, in the fact that it diffuses freely, it influences very close regions of tissue, rather than being confined directly to only one synaptic space. So in our last bit for this lecture, we're going to be talking about transmitter-gated channels and their comparison to metabotropic channels or G-protein coupled receptors. So transmitter-gated channels or ligand-gated channels aid in very fast synaptic transmission. They're sensitive detectors of chemicals and voltage, so voltage-gated channels and chemical or ligand-gated channels. They help to regulate flow of large currents, so allowing for certain ions to flow in versus out, and they're able to differentiate between similar ions. So the basic structure of a transmitter-gated channel, and all transmitter-gated channels mean is that they are specifically gated and they require a ligand or something like a molecule or neurotransmitter to bind, allowing for that channel to open. So the basic structure of these channels are that they are pentamers, meaning that they have 5 protein subunits, which come together to ultimately form a pore through which ions are able to flow. So an example of a pentamer, or an example of a transmitter-gated channel, is the nicotinic acetylcholine receptor. That specifically lies with the neuromuscular junction. So there are 4 different types of polypeptides that can be used as subunits. So you have alpha, beta, gamma, and delta. So remember that these are just different subunits, but the entire structure is a pentamer composed of 5 subunits. So simultaneous binding of acetylcholine to 2 alpha subunit sites is required for that channel to actually open. So in the brain, the nicotinic acetylcholine receptor is a pentamer, so it has 5 protein subunits, but most of the receptors are composed of alpha and beta subunits only, in a ratio of alpha 3, beta 2. So here's the generic structure. You have 5 protein subunits, alpha, beta, gamma, delta, In order for this channel to open, you need acetylcholine to bind to the 2 alpha subunits. Now, transmitter-gated channels, most of them contain 4 hydrophobic segments that span the membrane in subunits. So you have the nicotinic acetylcholine receptor, GABA A receptor, and glycine receptors. These are all pentameric complexes of subunits. So glutamate receptors specifically are tetramers that have 4 subunits that come together to comprise a functional channel. Now, in the glutamatergic receptors, the M2 region of the glutamate subunits, so here, again, note that this is glycine, GABA, and acetylcholine. Glutamate, specifically, the M2 region doesn't actually span the membrane, but actually forms a hairpin that both enters and exits from the inside. So there are similarities in the structures of the subunits for different transmitter-gated ion channels. And here we have the receptor identity, acetylcholine, GABA, A, and the different subunits that they compose. So note here, so GABA A, alpha 1, GABA A, beta 1, GABA A, gamma 2 subunits, there's a similarity in their structure. So these polypeptides for various channel subunits are stretched out in this diagram. So they have in common the 4 regions, again, the 4 regions M1 through M4, which are segments where the polypeptides coil and span the membrane. So amino acid-gated channels are glutamate-gated channels. We talked about 3 different classes being AMPA, NMDA, and kainate. And AMPA, NMDA, and kainate are the agonists to those particular channels. AMPA and NMDA channels mediate the bulk of fast excitatory synaptic transmission in the brain. And remember, glutamate is the major excitatory neurotransmitter in the brain. AMPA-gated channels are permeable to both sodium and potassium, and most are not permeable to calcium. Most are not permeable to calcium. AMPA receptors mediate excitatory transmission, where they allow for more sodium to flow in versus potassium out. And NMDA channels are specifically permeable to calcium, and they're voltage dependent. So NMDA-gated channels cause excitation by emitting an excess of sodium, but again, compared to AMPA, they are permeable to calcium. And this inward ionic current through the NMDA-gated channel is voltage dependent. So, highlighting the importance of calcium, presynaptically it's vital in order for snares to operate. This allows for the fusion of the vesicular and presynaptic membranes. Postsynaptically, calcium has the really cool ability to activate numerous enzymes, not only regulating the opening of a variety of channels, but it can also affect gene expression. So it also, as there's sort of like a double-edged sword with calcium, in excessive amounts, calcium can trigger apoptosis, which is cell death. NMDA receptor activation can cause widespread and lasting changes in the postsynaptic neuron, and a lot of these changes can lead to long-term memory as well as implications in learning and memory, which you'll definitely discuss a lot more in detail in 376. So the magnitude of the inward ionic current depends on the membrane potential of the postsynaptic cell upon which glutamate is acting on. So at normal resting potential, which is negative 70 millivolts or negative 65, whichever you're remembering, the channel becomes clogged by a magnesium ion, and this is known as the magnesium block, and magnesium is Mg2+. So when the postsynaptic membrane becomes depolarized through activity of AMPA receptors, allowing for sodium to flow in, the magnesium ion pops out of the pore. Now this usually allows the activation of AMPA channels at the same and neighboring synapses, and it allows for NMDA to bring in calcium. Now note, Mg2+, this magnesium has a positive charge, so if you're depolarizing the postsynaptic membrane, you're creating more positive, so there's an electrostatic repulsion, so that magnesium ion that is normally in the pore will be able to pop off. So inward ionic current through the NMDA channel is voltage dependent, meaning that the membrane needs to be depolarized in order for it to occur. But then in order for the NMDA receptor to be activated, it needs to have NMDA or glutamate bound to it. So this current is voltage dependent and transmitter gated. So both glutamate and depolarization must coincide before the channel will allow for current to pass. So here, you have the NMDA receptor and the AMPA receptor, AMPA in blue and NMDA in pink. At resting state, negative 65 millivolts, you have magnesium, which is blocking the pore. So even if glutamate binds right here, there's nothing that is happening because this magnesium is literally clogging the pore. Now, if you depolarize the membrane sufficiently, this magnesium ion will pop off, glutamate is bound, and this allows for sodium and calcium to flow and be able to flow through. Now, GABA-gated and glycine-gated channels are mediating synaptic inhibition. GABA mediating most of the synaptic inhibition in the central nervous system, and glycine mediating non-GABA synaptic inhibition. So both the GABA sub A, and this is the ionotropic or ligand-gated channel, and the glycine receptor, these gate chloride channels. And again, chloride is an ion that contains a negative charge. So if you're allowing for chloride to flow into the cell, you're creating more and more negativity. You're taking that membrane polarization and you're hyperpolarizing it. So here's your resting state of negative 70. If you're allowing for chloride to flow in, you are hyperpolarizing, taking you further away from your magic number of around negative 55 millivolts. So you're making it harder for that neuron to fire. Now, the GABA A receptor also binds other compounds like ethanol, benzodiazepines, and barbiturates, and this is why mixing alcohol and particular drugs or depressants are really, really dangerous. So when GABA is present and benzodiazepines are present, this increases the frequency of channel openings, allowing for more and more chloride to flow through. Now, when barbiturates are present along with GABA, this increases the duration of the channel openings, meaning that the GABA channel will remain open for longer periods of time, allowing for more and more chloride to flow through. This leads to depression, not in terms of the clinical sense, but you're depressing activity in the neuron. And if you depress it too much, particularly in areas of the brain that definitely need it, like the medulla, hindbrain regions, which are regulating autonomic processes, this can result in death. So the last little bit that we'll talk about is G-protein coupled receptors and effector systems. So there are three general steps in transmission of a G-protein coupled receptor. You have the binding of a neurotransmitter to a receptor protein. You have the G-protein that becomes activated. And then this G-protein will then go to activate numerous effector systems. So the basic structure of a G-protein coupled receptor is a single polypeptide with 7 membrane-spanning alpha helices. That means there are 7 alpha helices that will span the membrane. So the transmitter binding site is made up of 2 extracellular loops of the polypeptide, and the variations in the ways that these extracellular loops are can determine what is able to bind. So 2 of the intracellular, now note the intracellular loops, so on the inside of the cell, can bind to and activate G proteins. So structural variations here are determining not only which G proteins are active, but what effector systems are activated when the neurotransmitter binds. So here are some G protein coupled neurotransmitter receptors. So note, some neurotransmitters like acetylcholine and glutamate and GABA can have ionotropic as well as metabotropic receptors. So acetylcholine, muscarinic receptors, glutamate, metabotropic glutamate receptors. In GABA, GABA sub B. So the structure of a G-protein coupled receptor includes the G protein, which is simply GTP binding protein. That's where you get the G from. And a signal, so from the receptor to the effector proteins. So here you have your basic G-protein coupled receptor. This simply means that it is going to be coupled to a G-protein found in the intracellular space. When a transmitter binds and G-proteins are bound, you'll replace the GDP with GTP. Then the G protein will dissociate and affect other proteins, including effector proteins, channels, or enzymes. And then the GTP will go back to GDP, and your G protein will come back together. So in an inactive state, so inactive, meaning there's no neurotransmitter bound to this G protein coupled receptor, there are 3 subunits to the G protein. You have alpha, beta, and gamma. The whole complex, the whole G protein, this is the entire G protein. This G protein floats along the inner surface of the membrane, with alpha subunit of G protein being bound to GDP. Now, if the G protein complex bumps into the proper receptor, remember that they're intracellular coils along with extracellular coils, allowing for transmitter to bind and G protein to bind. Now, if the G protein complex bumps into this receptor with the transmitter bound, then the G protein will release its GDP for GTP that is found from within the cytosol. So activated G alpha will bind GTP. The activated GTP bound protein will then split into two parts. You have alpha, which is bound to GTP, and you have G beta gamma. So you have the G alpha subunit plus GTP and the G beta gamma complex. Both of these are able to move on to influence various effector proteins. The G alpha subunit of the G protein complex is in itself an enzyme. Now, alone, it is going to actually break down GTP into GDP, eventually terminating its own activity. So the G alpha and the G beta gamma subunits come back together and the cycle will be able to begin again. So one way that we can divide G proteins up is whether or not they stimulate or inhibit effector proteins. So you have G sub S, which are G proteins that will be roughly excitatory or stimulatory, and then G sub I, or inhibitory G proteins. So there are 5 steps in G protein operation. In the inactive state, you have 3 subunits, alpha, beta, and gamma, and this G protein complex floats along the inside of the membrane. So alpha is bound to GDP. In its active state, where the G protein will bump into the activated receptor, and this exchanges GDP for GTP that's found within the cytosol, and then you have a breaking apart of this complex. So you have G alpha, which is bound to GTP, and you have G beta gamma, which can then influence effector proteins, respectively. G alpha will inactivate by converting GTP to GDP, and then G alpha and G beta gamma recombine to start the cycle again. Now, there are 2 different pathways. You have the shortcut pathway, known as the membrane delimited pathway. This is the fastest of the G protein pathways, and it's very localized. So this is from the receptor to G protein, allowing for an ion channel to open. This is very fast and localized at the level of the membrane, allowing for an ion channel to open. So a good example of this shortcut pathway is found in the muscarinic receptors in the heart of the acetylcholine receptor, or GABA B receptors coupled by the shortcut pathways to potassium channels. So in the muscarinic receptors in the heart, these are coupled via G proteins that are coupled to particular potassium channels, which explains why acetylcholine slows the heart rate. So G beta gamma migrates laterally along the membrane until it binds to the correct type of potassium channel and induces it to open, allowing for potassium to exit. So here is one segment of the membrane delimited pathway which is occurring. Acetylcholine binds to the muscarinic receptor. The G protein, which is found in the intracellular side, bumps into the muscarinic receptor. Alpha bound to GTP, G beta gamma. G beta gamma will go and affect a potassium channel, allowing for it to open. The second effector system is second messenger cascades. So the G protein couples the neurotransmitter with downstream enzyme activation. So you have a neurotransmitter that binds to a receptor. This leads to alpha GTP, which can affect an enzyme, which can then create an activation of downstream secondary chemical reactions. So this is what's known sometimes as the push-pull method, where different G proteins can stimulate or inhibit adenylyl cyclase, creating cyclic AMP, which can then activate downstream enzymes, or, depending on the receptor subtype, can lead to an inhibition of this conversion, meaning no cyclic AMP. So here, in our stimulatory G protein, if you have norepinephrine bound to a stimulatory beta receptor, this can lead to the activation of alpha GTP, which leads to the activation of adenylyl cyclase, creating additional second messengers, which can then go and stimulate things like protein kinase. However, if you have norepinephrine that binds to an inhibitory alpha-2 receptor, or an alpha subtype, this can lead to a G protein becoming active. This will then act or inhibit adenylyl cyclase activity, preventing ATP from being converted to cyclic AMP. So back to this whole idea that depending on the different type of receptor that you are bound to, you're going to have different effects. So some of these cascades branch. So one example is when the G protein can activate phospholipase C, which can then generate diacylglycerol and inositol and IP3, which can then activate different effectors. So the activated G protein here, we have a neurotransmitter that binds to a G protein coupled receptor. G alpha bound to GTP can activate phospholipase C, which will then split PIP2, or phosphatidylinositol 4,5-bisphosphate, into two molecules that will be able to act in their own separate rights as second messengers, diacylglycerol and inositol 1,4,5-triphosphate. Now, IP3, again, both of these chemicals are derived from PIP2 and the activation of phospholipase C. IP3 is able to stimulate the release of calcium from intracellular smooth ER stores. That released calcium can then go on to stimulate various downstream enzymes, including calcium calmodulin-dependent protein kinase, or CAMK. Now, this has the ability to actually go and affect other enzymes down the stream. This is implicated in molecular mechanisms of memory and learning, which you'll definitely talk about more in detail in Psych 376. Diacylglycerol, from again this conversion of PIP2 through phospholipase C, can stimulate downstream enzymes such as protein kinase C, which can then phosphorylate different proteins down the way. So, the main difference between ionotropic, or ligand-gated channels, and metabotropic channels is, I hope, very clear at this point, because metabotropic channels are causing a lot of metabolic changes far down the cell compared to just directly allowing for a channel to open. So phosphorylation and dephosphorylation are very important ideas when it comes to protein activation. So phosphate groups can be added to or removed from proteins, and what this addition of phosphate or taking away of phosphate does is it actually changes conformations and biological activities of these particular channels or these proteins. So phosphorylation of ion channels can strongly influence the probability that they will open or close. And so when something is phosphorylated, you have a protein kinase that is activated. Protein kinases add phosphate groups. Protein phosphatases take away phosphate groups. So that's a very big, important distinction to know. Kinases phosphorylate, phosphatases dephosphorylate. So the function of signaling cascades allows for signal amplification. So activation of one single G protein coupled receptor can lead to the activation of numerous ion channels. So the transmitter can activate a receptor, which can then activate a G protein, which can then lead to the addition of more and more second messengers. This can activate enzymes, which can then go and phosphorylate other channels, allowing for activation or deactivation of those channels. So the process of divergence and convergence is readily apparent in these neurotransmitter systems, where in the instance of divergence, one transmitter is able to activate more than one receptor subtype, allowing for a greater postsynaptic response to occur. Whereas convergence is where you have different transmitters converging to affect one entire or to affect the same sort of effector system. So numerous transmitters affecting respective receptors affecting a system, whereas in divergence you have transmitters that can affect different subtypes going off to form a greater postsynaptic response by affecting other systems. So at this point, I hope that it's relatively clear, or a little bit more clear, that transmitters do a whole lot of stuff in the nervous system. I'm not trying to sound very brief or casual about it, but neurotransmitters are what essentially create our perception of the world and are what allow for chemical communication to occur. So transmitters transmit information between neurons. So in our presynaptic cell and our postsynaptic cell, this is a very large postsynaptic cell, you have your neurotransmitters that are released from the presynaptic to the postsynaptic, or from the postsynaptic to the presynaptic, and regulating information transfer. So this is an essential link between neurons and effector cells. So signaling pathways, our signaling network within a neuron, somewhat resembles the entire brain's neural network. So inputs can vary temporally, or based on time, and spatially based on space, to increase or decrease particular neuronal drives. And this is again delicately balanced, where you don't have too much inhibition or too much excitation, and then in certain clinical cases when you have too much excitation or inhibition, you're able to sort of understand why or how that's occurring when this balance is disturbed. And then signals help to regulate other signals. So drugs can actually shift that balance of signaling power between too much excitation or too much inhibition, or also too much neurotransmitter being released or not enough transmitter being released. So I hope that you've enjoyed this sort of whirlwind through all the transmitter systems. Definitely go through your book and make sure that you understand the transmitter systems in terms of synthesis and degradation. Go through this lecture that I've provided as well as this video a few more times just to make sure that you have everything sort of good and crystal clear, because on the test you're going to be asked to differentiate between a lot of these transmitter systems, and it can get a little confusing between all of the rate-limiting enzymes and the rate-limiting steps. But the more that you sort of marinate in this information and look at it and work with it, the easier it will become. So I'll see you all in Chapter 7.